Hello students, welcome back to Biotech world. So the CS Biotech is now going to teach you about biotechnology and in agri sciences. Means what is the role of biotechnological techniques in the agriculture sciences, including horticulture, animal breeding, and all that, including the crop yield, increase of crop yield, and everything. So we will talk about these factors in this lecture. There has been increased activity and research between different agriculture areas with common research techniques and goals. Means as I told you, different research techniques are used in different agriculture areas like plant sciences, animal sciences, environmental sciences, health, and agri medicines. So we will talk about these step by step. So first of all, plant science: wide scale production of transgenic plant impacting the horticulture. So we again use transgenic plants, like we use genetically modified organisms, GMOs. We inject the DNA of our interest in different plants, plant cells, and or plant seeds to grow our product of interest, like to increase the yield, like to increase the strength, to increase the immunity of the plant, so it can fight with the external environment in a better way. So these are different purposes that which are behind the use of biotechnology in plant sciences. The next is animal science. So while in animal science, increased use of methods of in vitro fertilization and artificial insemination improve selected breed programs. So what is selective breeding? Actually, we first have to know about this. You may have studied in your 8th class, 10th standard biology, that in vitro I was talking about selective breeding. So the selective breed means you will select the genes, you will select the qualities that you want in the upcoming generation, and you breed those animals which have those qualities, so the next upcoming generation will have those properties in common. And like if one cow produce more milk and you breed it with another one which is more immune, for example, so after crossing these two, you may get in the next generation more milk and more immunity, both. So this is selective breeding in vivo actually. But in vitro fertilization, we get semen of interest, and we get a egg from the mother side, and we do in vitro fertilization, and then later on it produces the offspring of the interest, that whatever the qualities that we want in that, we will get that in there, because the gene of interest is the main basic purpose, that we inject the gene that we actually want from that particular species. So in the next generation, we will get those properties. In environmental sciences, use of biotechnology techniques in the environmental science for cleaning contaminants and protecting endangered species. Bioremediation, use of natural organisms to clean contaminants. So in environmental sciences, we use different techniques to clean the environment, like to improve the environment for the living of different organisms that are about to get extinct, which are endangered species. Like we call endangered species which are prone to or which are near to become, what was the word? I just forget that. The ending of a species, endangered species are those which are about to end, which species no longer be available. I'm just forgetting that term that we use for this. Bioremediation, use of natural organisms to clean contaminants. This is another thing. So bioremediants means with living organisms they use to clean the environments. Like you can see, naturally, what's naturally working is we have bacteria which act on dead bodies and destroy and decompose them, and that decomposition produce some useful products that are later on used by the land, which can increase its fertility and some chemical occurrence, etc. So these similar techniques are used in biotechnology to clean the environment. Immunoassay tests are used to test for the presence of contaminants in soil, water, and even blood. Those who are in the field of biosciences, they all know about the immunoassay test. Like ELISA is a very famous immunoassay test. are used to test for the presence of contaminants in the soil. So we ELISA for humans or animals like blood test, but in soil and water we also do make some immunoassay tests to check if there is any kind of abnormality or presence of any contaminants in the installation of the biological barriers to prevent the transfer of harmful microorganisms between production facilities like tire wash channels. So we block different channels, so during the fertilization or during the production of a new offspring we get a better we get something clear and something better generation in the next one. Health and agri medicine. Farming, the creation of plants and animals capable of producing medical substances. So this farming start from PH, not F. The creation of plants and animals capable of producing medical substances, and the use of biological barriers to prevent the spread of harmful microorganisms that could contaminate food sources. These are very simple factors, so you can easily understand, but I can still explain that we produce specific barriers to prevent the spread of different harmful organisms in the next, which can contaminate the food sources. Okay, so DNA analysis, paternity testing has emerged as a technique to test the genetic ancestry of animals. So the problems, we will discuss problems in the later lecture. For now, this is all enough. See you in the next video. Goodbye.